Great. Just once again, welcome everybody. I just wanted to go through quickly the agenda. We're going to have about the first hour or so in the presentation, and then we'll have the Q&A in the last 30 minutes. So thanks for being here. And a big thank you to our partners at the University of Minnesota's long-term ecological research group. And without further ado, I'd like to turn it over to Emily Snellrud from the University of Minnesota. Emily, take it away.
Great. Thank you so much, Katie, and thanks for having us here. It's a really great opportunity to talk to everyone about some of the work that we're doing at Pig's Eye. And today we're going to introduce ourselves, tell you a little bit about who we are and why we care about Pig's Eye Regional Park. We'll talk about two research goals that we have, setting a baseline of sort of research knowledge at for our projects at the park, and then exploring the potential of biochar and bioremediation. And then at the end, we'll tell you briefly about some of our longer term research plans, and we would love to get input from the community in terms of what the community is interested in terms of research. So my name is Emily Snellrud. I'm a faculty member in the Department of Ecology, Evolution, and Behavior at the University of Minnesota. And I'm broadly interested in how organisms deal with human environments, why some are able to tolerate pollutants and others cannot, and what this means for restoration. And maybe we'll go in reverse order. Lindsay, do you want to introduce yourself?
Yes, hi, I'm a postdoc in Emily Snellrud's lab. My name is Lindsay Kemmerling, and I'm interested in biodiversity conservation and restoration and looking at through the lens of contaminants.
And I'm Lee Penn, and my name's Lee Penn. I'm in the chemistry department at the University of Minnesota. And broadly, my group is interested in removing a broad range of contaminants from natural waters.
Excellent. And I want to thank our funders for this work. We're working on 2 grants through the Institute on the Environment through their Strategic Goals program, a grant through the University of Minnesota's Office for Research and Innovation, and a grant from the State Environment and Natural Resources Trust Fund. All right. So Pig's Eye Regional Park is one of the largest parks in St. Paul with over 400 acres of land, and historically the home of the Dakota village of Kaposia. And Pig's Eye Regional Park is a beautiful site on the Mississippi. It's home to a lot of birds and reptiles, amphibians, insects, mammals, lots of wildlife and plants. And it also sits in a really important location within a network of green space in the Twin Cities and within a riparian habitat corridor along the Mississippi. So the Mississippi is an important flyway for migrating birds, and this green space along the Mississippi is important for migrating species as well. But Pig's Eye Regional Park has a history of pollution. It's the largest unregulated dump site in the state. There were several decades where there was unregulated dumping of industrial and a wide range of waste products. And there have been some efforts to clean up the site, but despite this, there's still pollution here today. There are over 8 million cubic yards of municipal, commercial, and industrial waste at the site, which you can see. We took this when we were out there in May. Even if a tree falls over, it will rip up pollution that's right under the cap of this landfill. So while it's an important habitat for wildlife, it is polluted. And there are a range of known contaminants there, heavy metals, including lead, cadmium, copper, and zinc. PFAS, historically other chemicals such as PCBs, and continuing today methane landfill emissions. So this map shows in brown where the city of St. Paul, their land ownership of the park, and then the dotted line, the approximate area for the dump site. And this impacts a range of communities, not just the communities in East St. Paul, which are directly adjacent to this site, but also everything downstream on the Mississippi, because this park flows into Pig's Eye Lake right down here, which then flows into the Mississippi. And then, of course, impacts on indigenous communities, being the historical site of Kaposia on Dakota land. So Pig's Eye is an important location. There are a lot of people that care about Pig's Eye. There are community groups like Friends of Pig's Eye Park, the Urban Bird Collective that do work on the site, monitoring what's there, helping to clean up the site. There are nonprofits like Great River Passage Conservancy, which care about the site. And then recently, the Minnesota Pollution Control Agency set up the Pig's Eye Dump Task Force to collect community input on the site and make a plan about longer-term cleanup for the site. So where do we fit within this community of people that really care about this site? We are researchers. We also care about this site. In working there over this year, have really grown to love this site. But we're also very interested in doing research at the site that contributes to not just restoration efforts at the site and improving the site, but also broadly contributing to restoration knowledge. So we're not necessarily in control over what the Pig's Eye Dump Task Force decides to do at the site, but we hope in doing some research there, we can inform broader restoration efforts at other sites in addition to Pig's Eye. So that's kind of where we see ourselves as researchers with respect to this broader community of people that really love Pig's Eye. All right, so what are we doing in terms of research? So we're gonna talk through two sort of broad research goals. The first is setting a baseline. And what I mean by this is kind of twofold. So first, we're interested in setting a baseline with respect to the pollution that's there currently. So the Minnesota Pollution Control Agency has been monitoring pollutants at the site for a long time. Some of these data are available for some online for some pollutants. So for instance, this is a map that they have for PFAS contamination at the site. You can see the pink wells here are ones that are above levels of concern for PFAS in the groundwater. So clearly PFAS is a major concern here. A lot of our research, at least in my lab that I'm working on with Lindsay, is less to do with water though, and more to do with terrestrial, so sort of the surface layer. Some of the work in Lee's lab has more to do with the water contamination. But we were interested in, well, what is at the surface level in terms of contamination if we're going to start to think about restoration for pollinators, possibly prairie restoration or terrestrial restoration. And there, the current data are less clear. So one of the first things we were interested in doing was setting out a map of surface soil contamination. And we did this by going out to the site in May and collecting about 300 soil samples. This is just at the surface of the soil, so the upper 10 centimeters of the soil. And then we used a rapid screening technique in the lab called PXRF to screen the site for hotspots of metal contamination. We were especially interested in lead, but we also took some data on other metals. And so here's a preliminary map of some of that surface soil contamination. And this map was generated by Nick Jelensky's lab, along with Nora Pearson and Morgan Smith. And in this map, and I apologize that it's red on green for colorblind folks in the audience, but in the background, you can see a satellite view of Pig's Eye Park. And the points here represent each soil sample that we took, these little circles. And the redder and bigger the circle, the higher the level of lead in that soil sample. And so there are a couple of things I'll point out here. So first, I'll point out that the EPA level of concern for lead is 200 parts per million. So right around here, this sort of grayish red color. And so you'll notice that a lot of the site, especially the areas where people are walking. So this is the parking lot to orient you here where people walk in the southern part of the park. Here's the trail out here. Here's Battle Creek. I'm assuming you can see my cursor. And then there's a main trail that kind of walks around here. And the good news is that in that main place where people are walking that primary loop, the levels are outside of that area of concern. There are some hotspots of lead contamination, especially down here in the southwest corner of the park, and then up here in the northwest corner of the park, where levels are getting up well above this EPA level of concern. And to kind of put this into context, this would be like concerning if you're sitting in the dirt, playing in the dirt, and possibly breathing in some of that dust, or accidentally ingesting some of that dust. It's not necessarily a concern if you're walking over here and the levels are elevated over here. But at least to us, with respect to research, this says that, you know, generally the areas in the park where people are walking around are safe, which is good. And if we're interested in doing some experiments in more contaminated soils, we might want to look up here or down in this part of the park. All right. So we also were interested in establishing a baseline with respect to biodiversity. And this is in part, so if there is a larger scale restoration, it would be great to have some before data. So before that restoration, what's there, but specifically numbers that we can compare using the same methods in the future after or during the restoration. We are also interested in what species were there and what species are common with respect to possibilities for species-specific research. So if we wanted to do any raising of butterflies on site or tagging of birds or something like that, what is there that we could work with? So we looked at both birds as indicators and also pollinators. And so we went out and before it started flooding this summer, we were going to do more point counts, but it flooded and we couldn't access the park. But we did a series of point counts where we went out and counted as teams, birds, the number of birds within a set radius circle. And here we're focused on what birds are actually there during the breeding season, actively singing, defending territories. So what birds are using the site as a breeding site. And for folks who have been out there watching birds, I don't think any of these will come as a surprise, but these are the some of the most abundant species that are there singing, defending territories, breeding on the site. So red-winged blackbirds above and beyond the most abundant, but also a few warblers, yellow warbler, common yellowthroat, song sparrows, and some other neotropical migrants, Baltimore oriole and warbling vireo. So the site, as we knew already from people doing work, watching the birds there, is providing really important habitat. And now we have some density numbers on there that we can compare to future surveys. All right, Lindsay, I'll pass it over to you.
Yes. Thank you. Okay. So I'm interested in insect communities, and butterflies and bumblebees are great species to study because they're of conservation interest. There's even some endangered or threatened species in the area. And because they can be surveyed visually. So you don't need to catch them, or you can catch them and release them. So this summer, I was trying to get a baseline of bumblebees and butterflies, and these are orange points on the map. If you've been there, maybe you can orient to where that is on the map, but I surveyed 100-meter long transects throughout the park. And I did this every other week from June to September. And yeah, I think that's what I say. Next slide, Emily. And these are just the species at the top that I found. So maybe you all have seen more species than this out there, but this was just along these set transects. Cabbage whites and common ringlets are on the right there. Those are the most common. And then for the open flowering plants, we saw both native and non-native species, about an even amount of each of those. And so it's good that there's some native species there, but we all know that there's a lot of non-native plant species out there too. And our goal will be to continue surveying these, one, if there's remediation efforts that happen, and after those, so we can continue to look at this long-term change. I think, yeah, so there's nothing to compare these results specifically to, but our goal will be to have this as a baseline to look at how this changes in the future.
Great, thanks, Lindsay. Okay, so our second research goal, we're interested in potential bioremediation at the site. And so what do we mean by bioremediation? So we're taking a pretty broad view to bioremediation and defining it as really any use of biology in any way to remediate a site. So what do we mean by remediation? Well, here we mean somehow minimizing the pollutant impacts on the ecosystem at the site. And this could either be by removing the pollutants that are there or binding them and making them less accessible to the plants and animals that are living at the site. So we're interested in a number of different methods by which you could bioremediate a site. And today we're going to talk about some of the current research that we're doing specifically using biochar. So what is biochar? So biochar is kind of like a fancy word for charcoal made out of biological waste. So this would be taking any kind of biomass. Most commonly, this is residue from forestry or agricultural waste. So say taking ash trees or oak trees that died from emerald ash borer or oak wilt, and then instead of letting that rot in the dirt, converting it into biochar. And this means burning it at a high temperature under low oxygen conditions. And when you make biochar, there are a number of potential benefits to doing this as opposed to just letting the biological waste rot in place. One of them, and this is how we were initially drawn to biochar, is that biochar has been shown in a number of studies to potentially bind pollutants. And that's because if you zoom in, so these are scanning electron micrographs of tomato biochar. If you zoom in really, really close, and here's the scale bar down here, these are 15 microns, so, or sorry, 50 microns, so 50 thousandths of a millimeter. So really small scale. There are these lots and lots of little pockets, little pores, and this high surface area and porosity of biochars can take up pollutants like lead and other metals, and also some other pollutants like phthalates. So depending on the chemistry, depending on the conditions in the soil, biochar has this potential to bind pollutants. And so this would be an example of mitigation that's making those pollutants less accessible to the things using that site, the plants growing there and the animals feeding on those plants. So this is why we were initially interested in biochar, but it turns out there's a lot of other excitement around biochar because biochar sequesters carbon for around 2,000 years. And the way this works, so normally if a plant dies and decomposes, it releases, as it's decomposing, it releases that carbon in its tissue back up into the atmosphere as CO2. But if instead you turn that biomass into biochar and just bury it in the soil, you end up keeping about 50% of that CO2 in the soil. It's basically locked up in this char. So there's a lot of excitement about biochar in terms of carbon sequestration as well. So we've been interested in biochar, potential applications of biochar in two realms. The first in the sort of terrestrial land realm, and then the second in terms of water pollutants. So with respect to the land at Pig's Eye, we've been working with Malini Srivastava at the University of Minnesota's College of Design. And Malini is an expert in making earthen materials, and she's been developing biochar-based bricks and plasters. And we got interested in possibly using biochar-based plasters at Pig's Eye and in restoration in general for a couple of reasons. So as Lindsay hinted at, and if people have spent time out there, you probably know that there are a lot of invasive species at Pig's Eye. So in this picture, you can see almost all of this grass is smooth brome. And so one of the restoration goals would be to shift away from these non-native species towards more native prairie species, if it were to stay a prairie. And so the first step in a lot of restoration would be, okay, now we need to kill the smooth brome. And so the idea is, instead of using an herbicide or plastic to kill the smooth brome, could we instead use a biodegradable plaster that blocks the light and kills the plants that are there? And then as it degrades over time, potentially improves the soil. So if it contains biochar, then it could bind the metals that are in the soil, making them less bioavailable, but also shift the chemistry of the soil away from nitrogen, which tends to favor non-native plants, and more towards carbon as you're adding this biochar in, which tends to favor native plants. So this is the idea, and this is something that we hope to be testing there over the coming years. At this point, we're in sort of the development phase, and at a different site, we have installed some test pilot plots with 10 different biochar plaster recipes. And so here's just an image that shows some of those different plaster recipes. They vary in hardness, and so this graph just shows these different 10 plasters ordered by ones that are really hard. So this was just taking a little penetrometer that you push on it and measure what force it takes until it pokes through that plaster. So these are really hard, and then these are not as hard. So the point is the recipes vary in how hard they are. And then we also see that the recipes, the plots that are harder, so this graph shows each point here is a different plot with different plaster. Those with harder plasters, after a month of installation, had fewer weeds poking through the plasters, whereas those that were softer had more weeds poking through. But I will emphasize all of our plaster plots had fewer weeds poking through than the control plots, which were just a thick layer of mulch. So the plaster seemed to be suppressing weeds, and we'll use this to choose a plaster to use in experimental plots. So I'll pass it back to Lindsay to talk about that at Pig's Eye.
Okay, cool. So we're interested in testing these biochar plasters at Pig's Eye, and then also combining them with different plant species that are known to uptake heavy metals. So here we have this potential experimental setup, and we want to answer the research questions. How does biochar plaster compare to other methods of suppression of these non-native species? And that's necessary before planting in new native species, so it's a part of the restoration process. And then how does remediation by different plant species interact with this biochar plaster to remove contaminants from the soil? So could a mix of biochar plaster and plant species do better than just the biochar alone? And then we can also test what plant species might perform the best at doing this. So our goal would be to start this in the spring. And we're working with St. Paul Parks and also Urban Roots. A lot of you probably are familiar with the organization Urban Roots, but they're an organization on the east side of St. Paul, and they focus on youth education and conservation and urban farming. I run an intro to research program with Urban Roots in the summer for some of their interns, and this is called the Science Deep Dive. So I'm thinking that with them, we could collect some samples from these plots in the summer, or we could replicate this experiment at one of the Urban Roots restoration sites. So students there could also collect data and ask research questions in the places that they're working and that they're really knowledgeable about. So overall, we want to test ecological questions that are directly applicable to Pig's Eye itself, but also hopefully beyond that. And then the overall goal is to support biodiversity and human health with these experiments. That's what I have.
Awesome. Thanks, Lindsay. All right, I'll pass it over to you, Lee.
Yeah, so my group is really interested in looking at how biochar might effectively remove some metals from water that is flooding into the site or what have you. And also testing the question, does the biochar hold on to the metals and not let them go back out into the environment? And so these are two things that we're really interested in. So the reason is, as the site floods and dries, then we see quite a lot of leaching of old landfill contaminants along the margins of Battle Creek. So the vision is to place pillows along areas where we have an influx of water that contains these contaminants. And hopefully the pillows filled with biochar will take up those contaminants and prevent their entry into Battle Creek or the lake or what have you. And this is all part of an Institute on the Environment, that's the INE Impact Goal Grant, which is a MnDRIVE environmental project. And the title of our project is Magnetic Biochar from Minnesota Invasives, Sequestration of Metals and Phthalates from Contaminated Stormwater. And we are a collaboration of a number of people, including folks from up north at the Natural Resources Research Institute, and St. Paul Parks and Great River Passage Conservancy. So the next slide would be great. So what is biochar? Emily already gave a bit of an introduction to that. The biochar that we're using is made using buckthorn, which is an invasive hardwood plant that we have to manage. And so you have CO2 from the atmosphere that's taken up by plants as they're growing through the process of photosynthesis. And then you shove that into a kiln and you do the pyrolysis in the absence of oxygen and you get charcoal. And what you see on the right-hand side of the image of the slide are scanning electron microscopy images of buckthorn biochar taken by a graduate student working on this project, Alex Castillo. And you can see that these pores, which actually run the entire length of the particle, are retained. And this is all carbon-based material. So just as we see a great opportunity for carbon sequestration, we can take this invasive species that has to be managed and remove it, and then use it for the benefit of producing biochar. The next slide would be great. So our vision is, if you look in the upper left-hand corner of this sort of schematic, is to characterize the biochar so that we understand what kind of material it is. And then we do sorption experiments. And what sorption means is how does the biochar take up the things that we don't want in the water? So phthalates and other additives to plastics, metals like zinc and lead, which are known to be important contaminants at pig's eye. And then a new direction for the group is looking at whether or not biochar can lock down microplastics and prevent their transport as well. So we characterize their absorption, and then in the lower right-hand corner, I have this biochar modification corner. And what are our modifications? You know, we might use other plant sources other than buckthorn. We might change the conditions in the kiln. We might have a little bit of oxygen come in. We might go a hotter temperature, et cetera. And then also a really important thing that we're trying to do is impregnate that with iron oxides. And that has some advantages that I'll get to in a moment. And so our idea is to go from the lab, make biochars in the lab that we understand really well, take them into the field and run some waters through a field column and characterize the water at the bottom and characterize the biochar in the column, and finally land on field trial pillows where we place the pillows and then we collect those up, characterize the ability to remove the metals or whatever else has been taken up, characterize the biochar itself, and then characterize downstream water to see if we are effecting a reduction in the concentration of the things that we care about. And the next slide is just some example data. On the slide, the left-hand side, you see 2 vials, and we have a model organic pollutant that we're using, which is simply a blue dye. And that lets us have a really easy visual of whether or not the blue dye is being removed by the biochar. And so if we expose the biochar to 5 parts per million blue dye, afterwards, the solution doesn't look as blue. And the graph shows that the concentration of blue dye is on the x-axis here. As I increase the concentration of blue dye, I'm actually able to take up more and more of the blue dye. So basically what the thing on the left-hand side shows us is that the buckthorn biochar takes up our model organic pollutant. And then on the right-hand side, I'm showing data on a little bit different of a format where we did experiments looking at removal of lead from contaminated water. And so starting concentration at just 1 part per million, which I think we all can appreciate, there's no safe exposure to lead level. As we go up to higher and higher concentrations, we are successfully removing over 90% of the lead from water once we get past about 5 parts per million. So the point is, buckthorn biochar also takes up our model metal pollutant, in this case, lead. If we go to the next slide, we come back to our schematic, and in the modification part, what we really want to do is throw in a bunch of iron and make a mineral called magnetite. And magnetite is magnetic. And the beauty of that is then I can hold the biochar in place using a magnetic field. So what you see in this photograph is our in-lab reactor. And what you see on the right-hand side is the biochar pulled out of water by using a magnet. This is the magnet not under the plastic boat, and this is where the magnet is under the plastic boat. And you can see that the biochar goes right to the magnet because of the iron oxide loading. So the thing that's really nice about that is it gives us an easy way to separate the material from water that might be holding on to the biochar. And also, it turns out biochar floats, which isn't the most convenient thing. So if we can load it with something a little more dense, it makes it a little easier to manage. And being able to take advantage of magnetism is also lovely. The other final advantage of impregnating this with iron oxides is it actually makes metal removal even better. And so we hope to have even better performance. And then I think that's my last slide. Yeah, I think that's that.
Awesome. Thanks, Lee. Okay, so we'll end by talking briefly about some of our longer term research plans, but then we'll have plenty of time for a larger discussion, Q&A, and we're really, really interested in what the community is interested in terms of research and how the university can interact with the community to answer some of those questions. All right, so a lot of our work sits within a broader program called it has a very long name, Minneapolis-St. Paul Urban Long-Term Ecological Research Program, called the MSP-LTR for short. And this is a program funded by the National Science Foundation that's really meant to foster long-term ecological research. So we're interested in using the Twin Cities as kind of a model urban ecosystem to study the interactions between the human layer and the ecosystem layers and feedbacks between all of them. And this is important because we're a big piece of this long-term ecological research is thinking about contaminants and how they move through urban ecosystems. But we're very interested in long-term research questions. So not just working at the site for a year, but potentially working at the site for 5 years or 10 years or decades. And so we're interested in the opportunities presented by restoration at this site to study restoration of a contaminated site potentially over decades. And then using this to answer a range of other questions. For instance, one question that we're really interested in my lab is why some species thrive in contaminated sites than others, and then thinking about, well, how might this change as the site is restored? How might the community shift? And I'll turn to this broader question that we acknowledged at the beginning of this presentation, and that our longer term hope for this research is not just with respect to Pig's Eye. We love the site and we want to contribute to restoration at the site, but we also recognize that we're not the only player at the site, and we're not going to determine how the site is restored, nor do we really have capacity to restore the whole site. That's a huge project. But we're hoping that our research efforts at the site can contribute to knowledge that improves the site and restores the site, but also as we publish as scientific research, can contribute to restoration at other sites all over the country. And I see in the chat there's already a comment about biochar being used in remediation at other sites, for example, in Seattle. So we can learn from those efforts, and hopefully the things that we iterate on at this site, like making biochar magnetic or making a plaster, hopefully that's stuff that we can publish and other people can learn from that in restoration at other sites. So we definitely see ourselves as a small piece of a much larger effort. So I'll put this up here, and this is our second to last slide. We're really interested in sort of the broader community interest in the research at the site. We're interested in knowing for folks who are on this webinar, but also beyond. So you can pass this on to other people in your community. What questions would you like to see answered in the short term, so in the next couple of years, but also in the long term, over the next few decades? Where can the University of Minnesota and academic partners contribute? Where do agency partners fit in relative to this academic research? So we're really interested in input from the community, especially as we start to think about the next phase of our grant proposals, which we'll be putting together in the next few years. So I'll come back to that slide, but I'll just end by putting up emails for myself and also Lee, and pictures of other folks on our broader research team, some of whom are in the audience. So if people have other questions for them, yeah. So I'll go back here, and we have plenty of time for questions.
Thanks so much, Emily and team. That was awesome. Really appreciate it. Now, here's the interactive part for all of you. If you have a question, feel free to drop into the chat, or if you want to ask your question live, please raise your hand and I can unmute you. I'd love to just start out with my own question, since I'm the meeting host and can do that. Just a kind of a basic question, especially for Lindsay and Emily. Was there anything that surprised you in your initial kind of field research in terms of that surveying last summer, whether it was something that a pollinator or a bird or plant species?
That's a great question. Yeah, I was surprised at the diversity of birds that were there. I think when I initially went to the site, I was like, oh, there are just lots of red-winged blackbirds, but there are a lot of other species there, and the summaries that I showed were a lot of the species in more of the open habitat, but even in the woods, there are a bunch of other species that didn't show up on that top 6 list. And so there really are a lot of species using that site. And so kind of the follow-up research question for me is, how are they managing in terms of the contaminants that are there? Are they accumulating contaminants? How are they doing health-wise? So I think it just opens new research questions. And the ones that are thriving there, how are they doing that? What kinds of adaptations do they have to cope with the pollutants that are there? Lindsay, how about you?
I have two quick things. First is that there was the big flood in the middle of the year, and that definitely shifted at least the abundance of butterflies day to day, which there's no like statistical evidence of that, but just visually. So I think as a site that floods, it's very interesting to think of looking at this site over time, just how the community changes, but also within a season with flooding and things like that. And then second was there was so many orb weaver spiders late in the fall that were just so fun to watch, and for all the things that they eat, like including butterflies. That was very interesting. And the abundance is like very impressive.
Thanks, Lindsay. I'm just going to call up a few of the cool questions that are in the chat. The first one is from Russ Stark. And Lee, I think this is a question for you and maybe another member of the team that we haven't heard from yet. But Russ asks, what do we understand about when a given amount of biochar is full of contaminants?
Yeah, so this is a great question because a big challenge in the research is figuring out an easy to detect marker of when it's full, because there are two possibilities. Once the biochar is full, it could hang on to the contaminants, making them less bioavailable and not really a problem if they're on lockdown. That sounds like a reasonable outcome. But the other possibility is that once the biochar gets full, if some kind of condition changes, it might release all of those contaminants back into the environment. And then we've actually accomplished nothing. So we have a collaborator actually, because one possibility is monitoring changes in the magnetic behavior. So once we load the biochar with iron oxides, we'll have a detectable, measurable magnetic signal. And if loading the biochar with various contaminants changes that signal, we might actually have a magnetic measure of when it's full. And our collaborator on that project is Josh Feinberg, who's over in the Earth Sciences Department and is a major expert in the magnetic character of nanomaterials, because all the iron oxides that we stick in this biochar will be quite nano in character. And so actually really lucky to be able to have that collaboration. Does that answer your question? We also have plans related to the plaster, because I'm really interested in knowing if the plaster pulls up a bunch of metals. We could ask exactly the same question. How on lockdown are those metals? And if they're not on lockdown, what kinds of things do we need to do to prevent re-release of those materials? Thanks for the question.
Thanks, Lee. Another question comes from Whitney Clark. Whitney asks if this research, whether it's, for the whole group or independent parts, shows that these methods could be really effective in remediation. Has anyone estimated how much it would cost to implement them at scale at Pig's Eye?
That's a great question. And I just unshared my screen to do some Googling. So one of the other comments was about, other people using biochar for remediation. And so we do have some estimates based on some of these other projects. And there was actually a different Pig's Eye task force meeting with the MPCA where they talked about an analogous restoration effort at Thompson Reservoir, where they applied 245,000 cubic yards of activated carbon amendment. And I don't know the price for that. I was just trying to Google that, and I think it's $30 to $40 million for that. So that's expensive. That was all water though. So we'd need to think about, it would be different because this is a mixture of terrestrial and water. And at least with the plasters, currently the plasters that we're using need some changes to the recipe to make them scalable, because the clay that we're using is very expensive. So one of our reasons for trying these different recipes is to try different clays that vary in how affordable they are.
Emily, one follow-up from Russ is, as I'm sure you've noticed, as other people have who've gone out there, the wood waste operation adjacent to Pig's Eye Regional Park is rather vast and is not buckthorn. I think it's probably mostly ash trees. But has there been any thoughts on using that adjacent wood chipping operation that's right there as a source for some biochar?
That's a good question. I can check out the links there. So I know the city of Minneapolis has a carbon sequestration program, and Jim Doughton has been running efforts to set up. They're actually building a large kiln in Minneapolis, right on the edge of the East Bank campus for the university. And the idea there is that there will be a flow of sort of wood waste from buckthorn, ash, oak, make the biochar, and then distribute it. I'm not sure what their distribution plans are, but so I don't know how the wood waste at Pig's Eye fits into that, if that's going to be part of that flow. And then there, I also noticed someone was saying there's a recent call about these wood waste grants, and so there's a lot of interest in biochar and doing research in biochar with respect to carbon sequestration and thinking about sort of the bioeconomy of the Twin Cities.
Thanks, Emily. And just a shout out to Randy. Thanks so much for putting the link to the Tacoma project in the chat. Also, Randy, just wanted to check with you. Do you feel like that you had another question about if there was a facility producing biochar in Minnesota? And I feel like Emily answered that a little bit, and so did Russ in the chat, but just wanted to make sure you got your question answered.
I think we could also mention the Minnesota Biochar Initiative. And then also the work happening up at the Natural Resources Research Institute up north. They're doing quite a lot of work with biochar too. And our collaborator, Brian Barry, is one of the folks involved in that. So there's quite a lot happening in the state.
Yeah, I'll just add to that, the Minnesota Biochar Initiative, they, I don't know if it's live yet, but they've been working on a new website. They're a huge growing network of people in Minnesota that are producing biochar, interested in sort of the biochar bioeconomy and also biochar research. And Minnesota is also hosting the National Biochar Meeting next fall. And so we're actually very interested in possibly having do a field trip out to Pig's Eye, but we'll see. What's next?
Lee, this might be a question for you. I don't know if you saw, there's in the chat from Paul, who is asking about so the pollution levels due to along the creek due to 3M dumping barrels, and about whether or not you think the project will delay the Pig's Eye task force recommendations. We might ask, and if you want to, we could potentially ask Paul to come off mute to ask that question just to make sure we understand it.
Okay, sorry, I'm reading that question.
I'm reading the question as well. It's a really good question. I have a little bit of experience working on PFAS and absorption of PFAS by iron oxides. There's a huge number of people who are working on this. I don't know about the potential for a delay.
I doubt we would delay their decision, but in part because, yeah, sorry, Lee.
No, please, Emily.
I was just going to say in part, I mean, we're not taking data on PFAS, in part because MPCA has been doing a lot of monitoring of that. And we've been taking more data on metals, in part because our historical work has been on metal contamination, and because that's more relevant to the surface terrestrial restoration that we've been thinking about. But kind of as we hinted at in the talk, I think we're doing research, and we'll learn some stuff that might have some relevance to the site, but is also relevant to broader restoration research. But I think that task force is they're gonna come up with their recommendations independent, I think independent of what we find, because I don't think anything is gonna be super big news to them with respect to what we find. But it might inform if of the, there are like 4 options for what to do with the site, one of which is, remove everything over the course of 40 years. Obviously, like what we learn there, if that's the route, what we're doing there is not going to apply. But if they decide to do something about keeping things in place, then our research is slightly more relevant to that space than thinking about like how could we restore the land that's there or the water that's there. If they do dump truck everything out, then we'll still have some data and can publish that, and that can inform other sites.
Another part of Paul's question deals with the application of iron specifically to the site. The iron was added to the sewer waste and piped over Pig's Eye land. And Pig's Eye Park friends see what might be this iron oozing out the creek bank after it floods. A lot of the dirt is red. I'm really interested in knowing more about that because we can characterize iron when we do the PXRF, which is X-ray fluorescence spectroscopy, and just tells us how much of each element is present. And it might be interesting to see how much iron is near these places where we might be running our tests, because it could actually affect things. The iron might actually lock down some of the metals, but then the question is, would they still be bioavailable? Because the iron is something that's important for a number of different organisms and plants. And so I don't have a good answer for how it might affect the test, but I'm really interested in more information because I think this is an important variable that I didn't know anything about. So if you're willing to send me a copy of that letter, I'd be grateful.
And just I know Paul is really Kathy. So Kathy, if you need any assistance with that kind of communication, back to Lee, just let me know.
Okay, Kathy.
Oh, and thanks for your comments, Josh, as well.
I'll also add that I know MPCA was involved in some amendments and mitigation along the creek and where the site abuts the lake, with some soil amendments. And I don't know if iron was part of that. I don't have that much information on that, but it would be really useful for us to get more information on the history of what soil amendments were done there.
Yes, please.
Just a follow-up question about timeline. The question was tied to, the Pig's Eye Task Force timeline, but I'm also curious about your own research timeline. A lot was laid out, both in kind of both sides of the presentation. How much will happen in 2025, like the upcoming field season? So, Lee, as an example, I know a lot will be developed in the lab first. Are we going to get all the way to like the pillow stage in 2025, or do you think that will be like maybe 2026?
I think that's a great question. I think flooding will be a big open question. I think, field sites always have their variables that they surprise you with. I would hope that we could actually place some pillows in 2025, but if we don't, 2026 is absolutely doable. But I'm hoping we can actually at least do some early stage pillow work in 2025.
Emily and Lindsay, I'm just curious from your perspective as well. In terms of do you anticipate, if there's not flooding, that most of this work will be able to be completed this coming field season?
Yeah, I think the idea is we'll put plots in starting in the spring and then monitor them for as long as we can. And so that could be months. It could be years, depending on what happens at the site. And we're we have a number of sites that we're working at in collaboration with community partners, and recognize there are many external factors. And we take data as long as we can. And then when something else happens to the site, then we publish what we have. So with our test plaster recipes, we've gotten interesting and usable data after a month. It would be great to get data several years down the road, because that's the time course at which these plasters are going to break down and move into the soil and bind the metals. And so being able to measure plants that are growing there, 2 or 3 years down the road, and looking at metal levels in the plants, depending on the plot treatment, we would love to do that if the site is there and not being dumped away or dump trucked away. Yeah.
Just as a follow-up from Brandy, and thanks, great timing for that comment, Brandy. What opportunities do folks here on the call today have to witness or get involved in some of these activities next year?
That's another great question. In terms of the biochar plasters, at the site where we're piloting the recipes, we actually did those layouts with community members, and that was in the Como neighborhood, in South Como. And so we haven't yet decided how we're going to scale it up. So there's a chance we might have some concrete mixers out there instead of doing it by hand. So there might be opportunities for volunteers to get involved, although that might also depend on where we decide to put the plots and whether the levels of metals there are at levels that are of concern. We don't want to put anybody at risk of metal exposure. So there might be opportunities for helping with installation. Certainly there will be opportunities for people to go look at the plots afterwards. We want to put up signage and so people can take a look at them. At the other site where we currently have plots, we even have a little QR code and people can like record observations of which plots have weeds coming up and stuff like that. So yeah, Lee, I know you have stuff with respect to the biochar pillows too.
Yeah, so when we're at the column stage where I'm going to build a column and I'm going to pack it full of biochar and I'm going to feed it water from Battle Creek or near Battle Creek or whatever, I think that we could definitely use a few hands on deck to help with that process, moving water around, packing the columns, maybe having more than one, having a small crew of people could really make that go much more smoothly, and probably be pretty fun.
Well, thanks. And for all of you listening, oh, go ahead, Lindsay, sorry.
I was going to add that for biodiversity surveys too, I know a lot of people already have a lot of knowledge and interest about things at the site too. So if you have interest in surveying things, visually doing surveys, then reach out to us too, because we could change protocols or have things monitored more regularly or looking at certain species if people are interested.
And for all of you, keep following and reading the Great River Passage Conservancy's e-newsletter and things like that. Anytime we have any demonstration events or volunteer opportunities, or we'll be sure to put the call out as well. Now let's talk a little bit about fire. Andy had a great question that he put in the chat about if the future restoration is a prairie or a meadow, is there a way prescribed fire could happen on the site, and how might that affect the biochar plaster layer?
That is a good question. I don't know, but we could speculate. Yeah, we've actually already started talking a little bit about the possibility of doing a fire treatment before the plaster goes down, in part just because the brome is so dense, wanting to get that layer on while it's knocked back. But I recognize this question is about fire afterwards. So our vision, if it works, is for this plaster to kind of degrade over time. It's clay, soil, grass, biochar, so like and sand. So the idea is it will break down and through bioturbation move into the soil. And so I would think that it would be kind of minimally impacted by the fire, by any prescribed fire that would probably just knock back the plants. And then the plants are going to be interacting with the soil in terms of the metal uptake. And so I would think that prescribed fire would probably not negatively impact it.
The prescribed fire goes through pretty fast, right? Because if it's kind of slow and there's a lot of oxygen around, one concern might be that you lose your carbon sequestration and that some of that carbon goes as CO2. Right, like when we use charcoal in a grill or like whatever. The prescribed fire, I think, usually runs through a field fast enough that you shouldn't have too much carbon loss from the plaster. I think maybe Brian Berry is a good person to ask about that because he's expert in biochar. I thought I saw his name on there. I think he might be here.
I see his name.
I did just
ask to talk.
For some reason, it's not letting me.
Oh, you're on now. I had to unmute you.
Oh, okay. Sorry, go ahead, Lee. Repeat that. Yeah.
The question is, if you have a prescribed fire running through a field,
Mm-hmm.
is there potential carbon loss from the biochar?
This
Or
is,
the
I mean,
biochar
this,
plaster?
yeah, perhaps. But I would imagine a lot of the carbon is encumbered in the plaster and not exposed to the oxygen during a burn. But I'm not sure how porous the final plaster product is.
Yeah, and I suppose if you had a surface layer that you had some carbon loss, we could characterize that. So we could actually run the experiment where we
Yeah.
place the plaster. We have a couple of plots where we place plaster in the field in the burn zone, and then we harvest the plaster after the fact, and we could characterize how much carbon loss there would be.
Yep. That wouldn't be too hard.
And then we could also characterize the hardness and everything else, and we would have quite a few measures of how well the material withstands that condition.
Yep.
That's
And
neat.
I should point out too, when you talk about hardness, buckthorn is pretty hard. There's
Oh, I
a
meant hardness of the biochar, not hardness of the plaster, not the hardness of the plants.
oh yeah, sure. I was gonna make a comment that about just the buckthorn biochar itself that pertains to its hardness. Um, so uh, ash, for example, is a little bit less hard than buckthorn. And counterintuitive, when you hear hard, you think dense, which is right, but you actually get these larger pores in the buckthorn biochar, which give it a greater overall porosity relative to some of the other tree species. So I certainly think it's a very interesting candidate for absorption medium, which I think is really underexplored. So just wanted to add that in.
I want to also mention that Josh Feinberg made a really good comment, which is that if we have oxidized iron oxides around, the fire running through might reduce iron and we'd end up with a magnetite enriched sort of situation. And so that's another potential impact on the biochars. That's interesting. Yeah.
Well, thank you. Other questions people have? We have another team member who's chimed in.
Melanie, I don't know if you want to say anything else about that, but talking about it in terms of fire retardation. So that's, yeah, that's interesting. I wonder what it would mean in terms of actually doing the prescribed burn.
Thanks. I just got permission to
Oh,
unmute.
good.
Yeah, I just wanted to say that there's, depending on the mixed recipe, there's a lot of studies on plasters and building applications for fire ratings and retardation and responses. So I think the carbon question is really interesting. It would be interesting correlated to a lot of literature that exists in the plaster studies already done.
I suppose a risk might be that the plaster might protect seeds.
Mm-hmm.
And so then they wouldn't be susceptible to damage during the burn. So ideally, I think we'd want to place the plaster after the burn.
Well, I think the question is about regular burns in like native plantings, which tends to favor the native plantings. So like the native species are adapted. So and that's why part of why we're questioning whether we want to do a burn before we put the plaster down. But that's something that we'd have to talk to St. Paul Parks about. Or a small burn, not the whole site, just a little plot.
See, there's a question from Austa in the chat about planting willows as a treatment option. That might be a Emily question.
I think that might be a Lindsay
Yeah,
question.
I can answer that. That
Thanks,
willows
Lindsay.
have been known to be plants that are good at phytoremediating, meaning that they take up heavy metals from the soil. And then I believe there's been work at Pig's Eye before, kind of looking at cottonwoods at least, and other species that could do this. So they just are for consideration of a species that we could use to see how well they do at this site. And has there been thoughts on testing the pollutant contents? That's a good question. We haven't tested any pollutants in plants on site. But it would be cool because trees have been there they've experienced, a lot of them have probably experienced a lot of the pollution. And I don't know if you could detect within tree rings over time how pollutants change, but it would be really cool to look at that. Or just in the leaves and how much they can uptake now. I think, I don't know what has been currently done, but it'd be good to get before and after some ideas of what species are uptaking more than others. And then another question, this is like not what you asked, but that we've been interested is what to do with those plants afterwards. So a lot of people that we've been working with in community gardens and things like that are interested in, for example, planting sunflowers, are known to uptake heavy metals, so they'll do that. And then a question is what to do with those plants afterwards. Where you don't want to put them in your compost if you know that they're full of heavy metals. So we've just played around with other questions that we could maybe ask with our research plots, but a lot of it is just not known as to what would happen. Okay, Lee's saying that tree rings have been asked. That'd be really cool if someone knows
That
how
would
to
be
do
cool.
that. Or microplastics, that'd be even harder, but would be cool. Especially because we could line it up with what, like, the history of the site. If you could describe the history of the site, like through tree rings and descriptively as what would happen. If someone can do that, let me know.
I
That
don't
would
know
be really
about
cool.
microplastics because they might be too big to make it up the pores, but
Not going into plastics, probably.
mm,
Josh, welcome to the meeting. Do you have something you'd like to add live?
No, I just now noticed I had the capability of turning on my camera. Thanks. I've been here the whole meeting, listening to the fantastic questions. Appreciate it. The tree rings are a great idea for looking at the sort of medium to long-term sort of environmental history at the site. You'd also be able to look at paleo flood histories based off of morphology in the tree rings too.
We do have
Can
a
you say
tree
more about the morphology about the tree rings? Like what might you expect to see in terms of pollutants, for example? Like what happens to the tree rings? How do they look different?
In terms of floods, what happens is if you have a tree sitting in standing water for any appreciable amount of time after a spring flood, it'll actually start to affect the structure of the phloem and xylem rings that are actually being created within the wood. And so there have been other researchers here at the University of Minnesota that have looked at these sort of flood-induced structural changes to piece together flood histories in places like the Red River Valley through time. And yeah, that's something that could be done. That doesn't have anything to do necessarily with pollutants. Those are getting incorporated as trace elements within the woody material itself.
Thanks, Josh. I know we have at least one tree ring person tangentially involved in the urban LTR, so that's given that some of the higher lead levels are in the forested parts of the park. That would be really interesting to think about more.
I think you could do PXRF on, I mean, maybe not PXRF, but like you can excite the tree rings. Like you can put it into the scanning electron microscope and then you have an electron beam that's a much
That would be cool.
smaller spot size.
Yeah,
And then you should be able to get lead loading, like lead to carbon ratio as a function of position in the cross section of the tree. I think that
cool.
would work. That's like solid microprobe type fun times. Yeah, that should work really well.
It would be cool to know, yeah, how much things are still uptaking. I'm sure there was a big pulse before, but has it lessened over time?
Other questions? Please feel free to write them in the chat or request that you become off mute if you'd like to ask them live. I did put this in the chat, but this meeting has been recorded and we will definitely make sure to put it out on the website, and we'll send a note out about that. So I hope that everyone here can share broadly with your colleagues and friends who might be interested.
Great.
Well, I'll just ask our presenters, any last comments you'd like to make or questions you'd like to pose to the group before we start wrapping up today?
I'll just say thanks for the opportunity for sharing some of what we're doing at the site. As I mentioned at the beginning, I've really fallen in love with Pig's Eye over the last year, and I really am excited about what we can do there in the coming years. And if there are questions, research questions, just things people are wondering about, like, oh, I wish we knew this about this organism or this plant or this metal or whatever at the site, we would love to hear what those are. And I hope there will be an opportunity to put them in the follow-up form that Katie will send out.
Yep, will do. Definitely.
Thanks.
And I want to acknowledge not only the research team, thank you so much for being here. I'm really grateful that Urban Roots introduced us last summer. That was just very fortunate. And thanks to everyone here. I am sure that many of the people here have a long, deep history with the site and have been caring for the site for many years. And I think all of us share that together. And so I just want to acknowledge everybody here who's been really trying to take care of this place, particularly Pig's Eye Friends, over the past many, many years. So thank you for that. And I hope that we can all see each other next field season when we can actually see some of this awesome research manifest itself out at Pig's Eye Regional Park. And thanks, of course, to the City of St. Paul, particularly the Parks and Recs Department, who's just been a great partner on this project.
I'll just add that anytime there's a presentation where we're leaving with new experiments, that's like awesome, right? We already have like 2 new sort of things that we're going to do with this fire plaster connection and the possibility of tree ring work. I mean, I'm like leaving the meeting even more excited about the research than I was when we started, which is, thank you for that.
Wonderful. Well, thanks again, everything. I think we're at the end of our meeting today. And again, we'll be sending out a little bit of a survey. Please take that. We'd appreciate it, along with a link to the meeting. So great. Have a great rest of the day, everybody.
Thank you so much.